Yeah, it's kind of confusing. I was just too lazy last night, so I'm wearing glasses today. Okay, so it is now 4:45. It's 4:45 where I am, but 3:45 if you're in CST. So I'll just get started. So let me share my screen. Wait. Technical difficulties, please wait a minute. Okay, so the problem is I can't share screen because I'm not co-host. So I'll just wait, I guess, for a second. Wow, we always have technical difficulties when it's my turn. Yeah, this is a webinar, which just means that it's like a conference kind of, I guess. I don't know if the other person is here, so do you have any questions from last week while I wait for a second, I guess? I know some of you might have not been here last week because we had some difficulties, but any questions from that? So okay. I don't know if the monitor is here, so I might as well just like copy and paste the presentation link, just because I don't know if this is gonna work. So if it comes to like the worst case scenario, I'll just like talk through it while you guys can look at the presentation, because screen sharing is disabled for me. So fun. Okay, so there's a link if you want to just take a look through on some of the stuff we're going to be covering today. Okay, you know what? I'm sending an email. I don't know what to do. Okay, you know what? I really feel like the other person is not here, so I'm just gonna talk through it or try my best to. Wow, I love having technical difficulties. Okay, so let's just start from the first slide if you're following along in the presentation. Unfortunately, I can't share my screen, so I'm just going to try my best to like talk through it, and hopefully we'll figure it out further along. But for now, let's just start from the first slide. So today I'm going to be talking about DNA and DNA processes because, again, this is part of genetics and heredity, and honestly, I'm just teaching whatever I feel like teaching. Sorry. So if you weren't here last week, hi, my name is Brenda. I am a rising freshman from Michigan, and I am teaching heredity and genetics because when I was in middle school, I was part of the Science Olympiad team for 2 years, and so one of the events I did was heredity, so that's what I'm teaching. Anyway, moving on to the second slide, I included a link to last week's presentation if you weren't here because we were experiencing some technical difficulties. So after class, or if you feel like it, just take a look if you want to learn. Last week we talked about Mendel's laws and also Punnett squares and that kind of thing. So on the third slide, the one with the diagram, this is a diagram of a eukaryotic cell. And this is not that relevant to what we're learning today, but it helps if you have like an idea of how the structure it looks like, because I'm going to be talking about things like located in certain parts of the cell and like moving around. So it helps if you have like a bit of an idea. So taking a look at it, this is called a diagram of a eukaryotic cell, and eukaryotic means multicellular. So that would be something like humans, plants, animals. We have multiple cells, so we have a lot of cells, like billions actually. But the other, like the opposite of eukaryotic is prokaryotic, which just means single cell. And single cell organisms have like a slightly different structure, but we're not going to be talking about that today. So, but if you're curious, feel free to like search up or something. So, taking a look at this. Oh, by the way, prokaryotic cells, like an example of them, I guess, would be like bacteria because there's just like one cell. And maybe you've heard of like amoeba. They're just one cell and the structure is slightly different. Okay, so taking a look at it, I can't annotate. Oh, okay. It would be really nice if I could annotate, but I can't. So I guess try your best to follow along. So the things I'm going to be focusing on today are ribosomes, which are like the tiny black dots. You can see them like around, like slightly around the edges of the cell, and also on what like those pink line things. This is difficult. So you can see lightly dotted on there. That's the rough ER, which stands for Endoplasmic Reticulum. Again, we're not concerned about it. I'm only concerned about the ribosomes. So ribosomes and these other things that are floating around in the cell, they're called organelles. And organelles are just like tiny things in the cell that have specific functions. It's like in you have like specific organs, right? You have a brain, you have like eyes, ears, that kind of thing. And they each have a specific function. So organelles are like organs. They all have specific functions, but they're tiny and they're inside a cell. The next part that we're going to be focusing on is the nucleus, which is like the red and pink thing in the center. Mostly just Yeah, the red and pink thing in the center. And the reason why that is relevant is because that is where all the DNA is stored, which is today's topic, so that's why it's important. Anyway, and lastly, the kind of yellowish greenish thing in the background, I guess, that's called cytoplasm. And it's in every cell. It's kind of like a watery gel-like substance, and it just like is in there so that they can have moisture, and also so like the organelles don't like keep crashing around each other. Anyway, I think that's it. Do you have any questions about like the specific parts of this diagram? We're really not that concerned with much of it. Honestly, I don't know what a lot of them do because this isn't really relevant to today's topic. Okay, no questions. All right then. So moving on to the fourth slide, what is DNA? So DNA stands for deoxyribonucleic acid. And that is why we abbreviate to just DNA, because that is a very long thing to say. So moving on to the fifth slide, DNA is the genetic code of all organisms. So DNA would code for specific traits you have, like your hair color, your eye color, like facial features, that kind of thing, maybe height. It's a bit more complicated, but like you understand what I mean. And it's common in all organisms, including like animals and plants. So they have like the same structure and the same base pairs. I'll get into that. So you can see on the right, I have a simple diagram of a strand of DNA. You can see that there are two solid strands, like two solid lines that are swirling around on the sides, and that is called the backbone. So DNA has a double helix structure, which means that it is kind of like twisted. You can see it, right? So it's like a twisted ladder. If you had a ladder, you twisted it and looked like that, and that is the structure. And the strands run anti-parallel to each other, which just means they go in opposite directions. So in math class, you might have heard of like parallel lines. They're just like they never meet. But anti-parallel would be they never meet, but they're in opposite directions. So one goes up and one goes down, or sideways. But yeah, they just go in opposite directions. And the next part of DNA, like the structure of DNA, would be nucleotides. So nucleotides are actually what makes up like the individual, what makes up DNA. Okay, so the smaller subunits of DNA would just be nucleotides. And that leads us to the sixth slide. And there was an animation on this slide originally, but oh well. So we're going to talk about the composition of DNA, which are nucleotides. So that photo looks really blurry. Oh wait, it's not that blurry. Okay, it just looks blurry when I look at it because my screen's kind of minimized. Anyway, so just taking a look at the diagram, we can see that there is a phosphate group, which is a little pink circle, and a pentagram, purple pentagram, and that's labeled pentose sugar. So usually we group those together. I'm not going to go too in depth into what they do, but we group it together and we call it a sugar phosphate backbone. So going back to the fifth slide with a diagram of the DNA strand, the yellow like backbone, that is all made of sugar and phosphate. And the reason, oh, back to the sixth slide, the reason why it's labeled as a pentose sugar is because, you'll see later. We'll see later. Okay, so, and then the last thing with the nucleotide is the blue rectangle, which is the nitrogenous base. And that is what I'm going to be going more into because, like, that's important. So, nitrogenous base, there are 4 in total. There's adenine, which we abbreviate A, guanine, which we abbreviate G, cytosine, C, and thymine, T. So, in the DNA diagram, you saw that like there were strands, but then there were like 2. You know what? I'm gonna go back to the slide. Back to the fifth slide. You can see that there's like the rungs of the ladder. They're kind of have like 2 different colors, you know. And if you were to take a closer look at it, you would notice like in this diagram specifically, the green always pairs with the purple, and the blue is always with the red. And that is because back to the sixth slide. Certain bases always pair with a certain other base. So, for example, A, adenine, always pairs with T, thymine. And a trick to remember, I guess, that my 6th grade science teacher taught me was apple in the tree, okay? I don't know if that's too helpful. I never remembered it like that. I just saw like at, and then the other 2 were paired together. I don't know, it might help, it might not help, but just remember apple and the tree. So A and T go together. And then the second pair is C and G. And to remember that, you could say like car in the garage. Again, I don't know if like these tricks really help, but if they help you, go for it. So looking at the example, I have like a for example thing. Usually I would have I can't annotate on this. Okay, this is frustrating. Okay, so I have a random DNA strand, so half of the DNA strand. Since there are 2 strands in DNA, I have half, like one strand, half of it on the top, and that is GTA, ACC, GTG. And the reason we separate it into like segments of 3 is because they're called codons. Later on, it'll make more sense, but just know that whenever we write out DNA or DNA or RNA, get into later, we write it out into groups of 3. Sometimes you might see it all just into one string, but like that's really complicated to read, and it's a bit of a mess. So just like go with groups of 3 for now. Okay. So since we have this strand, we know that G pairs with C, T pairs with A. So we just go down and pair each one to find like the complementary base. Complementary is like the one that pairs with it. So looking at the first one, GTA, G pairs with C. So the first one is C, then T pairs with A, second one is A, and A pairs with C, so the last one is T. So GTA, the complementary strand, it's like a segment, honestly. Complementary strand would be CAT. It would be a lot easier if I could annotate, but I can't. So do you guys understand like how the complementary bases work? Because I'm not gonna say it all. Oh, pretty cool. Yeah, that's a good way to remember, actually, if it helps you. Okay. Anyway, the next slide, slide 7, was supposed to be a practice slide, but since I can't screen share and I can't annotate, and I also can't exactly do like the animations I would click in and like the answer would appear, I'm just gonna talk you through it, I guess. So I gave you this strand of DNA, which was CGC, TAA, CGA, and it is in blue or purple. I don't really know what color it is. Indigo, maybe? So I have a little reminder on the right side, C pairs with G and A pairs with T. So using that, we would just find the complementary base for each of these like individual bases. For example, just for the first codon, which is the group of 3, C pairs with G, G pairs with C, and C pairs with G, which I already said. So CGC, the complementary strand, would be GCG. And the same applies to the other 2 codons. And do you have any questions on this, first of all, before I move on to the random facts? Okay, I don't see anything in the chat. Oh, by the way, feel free to use the chat or the Q&A option to ask any questions or anything like that. And so moving on to the random facts part, I only thought of this because I was writing out like the acronyms. They're not really acronyms, but like the reminders, the tricks to remember like A and T and C and G. And I remembered this thing. This is not as relevant for you guys because it's a little more complicated. I actually never knew about it until I was actually doing Science Olympiad. So I learned about heredity and whatever first in 6th grade, and then I did heredity as an event in 7th grade and 8th grade. So we never learned about this in 6th grade, and I just like suddenly found it in 7th grade. I was very confused. So A and G are purines, and that just means they have 2 rings in like the actual chemical structure of it. I'm not too sure what it actually means. I just know they have 2 rings. And you can see like on the bottom, I have a bit of a diagram. You can see that purines have 2 rings. Pyrimidines, which are the other thing, have 1 ring. And it's relevant sort of because a purine will always pair with a pyrimidine. So if for some reason you don't remember the base pairings and you do remember the purine and pyrimidines, then you could like sort of figure out how it goes if you somehow forgot. I feel like that's the only way it would be relevant, but oh well. Okay, so A and G are purines, which are 2 rings, like I said, and a way to remember this would be pure as gold. So purines, pure and then as called A and G. Just as a reminder, these do not pair together. They're just the same or similar chemical structures. And then C and T are pyrimidines, which means they have one ring. And something to help remember this by would be cut the pi, but pi is spelled like PY for pyrimidines. And CUT, the C and T represent cytosine and thymine. But there's a U, which leads us into our next topic. But before that, any questions? I know this is a very unconventional method to teach, but gotta do what we can do, you know? Okay, I'm gonna take that as no questions. So moving on to slide 8. What is RNA? So I briefly mentioned it earlier. Sorry, I was scratching on the side. So slide 8, what is RNA? RNA stands for ribonucleic acid. And you can see that there is a difference between like the D and R, obviously. So DNA, remember, it was deoxyribonucleic acid, while RNA is only ribonucleic acid. So it just means that there's no like deoxygenated sugar. This means there is oxygen in the sugar. That's not really relevant, just like, I guess, extra facts. So some things to know about RNA, because RNA and DNA do have differences. First of all, RNA only has one strand. So earlier I said that DNA had 2 strands and each of them are complementary. RNA doesn't do that. There's only just one strand, okay? And instead of thymine, you have uracil, which is why earlier I said like cut the pie, you have a U. So U now pairs with A, because you're replacing thymine. So for RNA, think C and G, still car in the garage, and A and U. And I don't know what to say about that. My teacher didn't give me anything. Just remember it's A and U, I guess. You're replacing T with U. T doesn't exist anymore, and you're going with A and U. So the other bases stay the same, which are A, C, and G. And RNA can travel outside the nucleus. So at the very beginning, I said that DNA always stays inside the nucleus. And that is because the nucleus is kind of like the brain of the cell. It tells like the cell how to function and whatever. So all the DNA stays inside there. However, RNA can go outside, and it'll come up later. Anyway, the most common types of RNA, because there are actually multiple types of DNA too, but like the most common one is DNA in itself, the one I just talked about. But in RNA, you will hear about multiple types of RNA. There are actually so many, but these 3 are the most common, and they're the ones that are relevant today. So the most common types are mRNA, tRNA, and RNA. And before we move on, which I'll be talking more about the different types of RNA, you can take a look at the diagram at the bottom, which just shows you like a visual comparison of DNA and RNA. So DNA, 2 strands, and you can see like the bases, they're listed on the sides, and RNA, 1 strand, also has the bases listed. Moving on to slide 10, which is types of RNA. The first box I have is mRNA, which stands for messenger RNA. And it is involved in transcription, mostly. That'll come up later. I don't think I made this presentation too well, but it will make sense later. So all you need to know is that mRNA is messenger RNA. It's created in the nucleus, and then it travels outside the nucleus through like the cytoplasm to a ribosome for translation. Oh, I guess I never fixed that. Wait a minute. Sorry. I was making this presentation really late at night last night, so. The next type of RNA is tRNA, which stands for transfer RNA. And transfer RNA is mostly just involved in translation. So mRNA is in transcription, tRNA is in translation. And tRNA brings amino acids. This will all make a lot more sense later. I just need just giving you a brief overview, just like something to keep in mind. And the last type is rRNA, which stands for ribosomal RNA. So this just sits in the ribosomes. It's mostly, makes up 80% of like what ribosomes are made of. If you remember, ribosomes are like those little black dots on the diagram at the beginning. If you need to go back and take a look, feel free to do so. And it is the site of translation, which will make sense later. Okay, so moving on to slide 11, a quick recap of DNA versus RNA. DNA is double-stranded. RNA is single-stranded. DNA has thymine. RNA has uracil. DNA always stays inside the nucleus, and RNA can travel outside the nucleus or just like simply exist outside the nucleus. Any questions so far on RNA? I know that was kind of confusing because I didn't really explain like what translation and transcription are. That is what we are getting into next. No question? Maybe. Okay. I'll take it. Okay, so the last section, the third section, is processes of DNA and RNA. And there are a lot of processes, but I'm just going to be going over DNA replication and protein synthesis, with protein synthesis happening first. So as a disclaimer before I go into this section, this is by no means like all of the steps that are involved in these processes because they are very, very complicated. And I honestly do not understand the entirety of these processes. I just know like the basics because that's what I was taught. But these can get very complicated. So I'm just going to be going over the very basics because if I get any more complicated, frankly, I don't know how to explain it. And also, it's just really complicated. Okay, slide 13, protein synthesis, which is going to be the first thing I'm talking about. So protein synthesis happens in two steps mostly, transcription and translation. And you might remember that from the slide where I was talking about the types of RNA. So transcription is from DNA to mRNA. Okay, I guess the names of these steps are kind of confusing. It honestly took me a while to like figure them out. So transcription comes first, translation comes later. So first transcription, we have DNA inside the nucleus, and then we create, not we, but like our mRNA is created. So using a strand of DNA as a template, the mRNA comes in and it fills in like the complementary bases. Okay. And since DNA is double-stranded, one of the strands will be used as a template and the other one will not. Moving on to translation, I have a diagram on the next slide, so this is slightly confusing, but hopefully it'll make more sense later. The second part is translation, which is where we go from mRNA to proteins. So proteins are, I guess, like chemicals. Okay, proteins are the things that like literally make up everything in your body. So like your hair, skin, it's all made of proteins. And that is why it is so important. That is also why it is coded from DNA. Because remember, DNA has all your genetic code. So using mRNA and other things, it's read, and that creates the proteins which make up everything. Okay, so proteins are made of amino acids. And there are around like 500, maybe 200 or 500, 200, 500 amino acids in like existence, but the only ones in the human body, okay, only 20 are in the human body. The other, I don't know how many, there's a lot, are in plants, animals, that kind of thing. So after the mRNA is created from that DNA strand, it leaves the nucleus, goes into the cytoplasm, and it finds a ribosome. So at that ribosome, there is rRNA. If you remember, that's what makes up the ribosomes. So the rRNA reads the mRNA, and by doing this, it will create like, it won't create, but it'll call for complementary strands again. So that's where tRNA comes in. So it's kind of confusing, especially because I can't annotate, but we have the RNA, right? The mRNA comes in, it's read by the RNA because something needs to read it. And so the RNA then calls, they don't really call, but then the tRNA come in, which are again transfer RNA, and they bring the amino acids to the ribosome, and they're strung together. So that is like the basics, I guess, as well as I can explain it. So let's take a look at the diagram because honestly, it is confusing. Okay, so hopefully you guys are on slide 14. and this is the diagram of what happens during protein synthesis. So I'm going to zoom in because I can not read this. Okay, so starting with DNA at the top, we can see that mRNA transcription happens. So you can see that the DNA kind of like splits apart at a segment. The mRNA comes in, fills in the complementary bases, then it leaves the cytoplasm to go for translation. That is on the, I really wish I could take it. It's transported to the cytoplasm and to a ribosome for protein synthesis. And you can see that the kind of like the gray blotch in the background, that is a ribosome. I don't know why it looks like that, but it does. So the mRNA is the twisty thing that's going through the gray splotch, and the weird bendy things, I'm not really sure what to call them. They look like beakers almost, but with like knobs on each side. Those are tRNA. So you can see at the top, they carry like a little dot, and that is an amino acid. So in the diagram currently, you can see that there is a chain going on. So they come in, they match with the complementary bases, and then the amino acids, they just like connect together, and then the tRNA leaves. I really wish I could, okay, so think of it like this. There's the mRNA. The tRNA comes in with like the complementary bases and with an amino acid. One comes in, that's like the starting point, I guess. And then I don't have multiple hands. Okay, so one comes in, and then another one does, and the two amino acids link together. And as a third one comes in, so that the second and third amino acids can link together, the first one leaves. And that is how it eventually forms a chain. And these chains can be very, very long because obviously the human body is very complicated. But after all the amino acids are linked together and it forms like a chain, that is what we call a, I actually forgot what it's called. Okay. Okay, it forms, oh yeah, it's called a polypeptide chain. Okay, so this very long chain is called a polypeptide chain. And then it folds together in a very specific way, which creates a protein. Then the protein does everything else in your body. Okay, like just gives you like hair and skin and whatever. Any questions about this? This is very confusing, especially because I can't draw. So any questions on this so far? Is there any way I can like summarize it? Okay, I'm gonna summarize it really quick. So we got transcription and translation. So it just goes from DNA to mRNA to RNA. Well, RNA is kind of like reading. It's not really involved. Okay, DNA, mRNA, tRNA, and then protein. The rRNA would be in between the mRNA and tRNA because it's reading the mRNA. Anyway, moving on to slide, yeah, I know, I'm not sharing screen because unfortunately I do not have permissions to share screen because technical difficulties. So did you just arrive, or, because I can just copy and paste the link for you. Let me do that. Okay, so unfortunately I cannot share screen. So I'm just talking through the presentation that I have. I don't think the other person is here, like the actual host. I don't think she's currently here. So unfortunately, I just have to talk through it. It's rather difficult, but hopefully you guys are understanding. Okay. Moving on to slide 15. This is where I have an example of how protein synthesis would work for like a imaginary strand. It would never be this short, but here we are. So I have an example on slide 15 just to show you how it might work. The DNA strand we're given is GAT, CGG, AGT. Then the next step would be translating it to mRNA. Well, transcribing it to mRNA. It's just transcribing it to mRNA because remember, DNA first, then mRNA, and then it, like the protein synthesis happens. So again, the same base pairing happens, C with G, A with U, remember, because this is RNA, and still T with A. which is where it gets slightly confusing, because if you see a T in the original DNA strand, it still goes to A, but if you see an A, it goes to U. Slightly confusing, but if you do it a couple times, you'll get it faster or more easily. The next part would be turning the mRNA into tRNA. So I guess I kind of, it's so confusing. Okay, so tRNA, you remember, like, go back to the diagram, I guess. Go back to slide 14. You can see the little weird glass beaker with knobs thing. You can see that they have like the rectangles at the bottom. Those are the complementary bases. So this is why earlier I said we separate DNA, RNA, and that kind of thing in groups of 3, because tRNA is in groups of 3. So every 3 codons codes for a specific, every 3 bases codes for a specific amino acid, which is why we separate in groups of 3. Back to slide 15. And so because tRNA needs to like match with the complementary bases, so RNA reads the mRNA. Oh, this is so confusing. RNA reads the mRNA, and then it will call for like the complementary bases for that mRNA strand. So then the tRNA, they're flying around and they're just waiting until they can come in, because they need to be complementary. So in this example, the first codon is CUA. So the RNA reads it, understands that it says CUA, and calls for a tRNA that is complementary to it. So in this case, it would be GAU. The same applies to the rest of the strand, the other 2 codons. And what's interesting is you notice that the DNA and tRNA strand are exactly the same, except T is U because it's RNA. So in a lot of tests, if it just asks you, give you the DNA strand and then just like straight up asks you to list the amino acids and the protein, you can usually skip the mRNA step and just directly copy down the DNA strand, but turn every T into a U instead. However, other times tests will ask you to like first transcribe it into mRNA and then translate it into tRNA, which is why we're here. So it's just an interesting thing to see, because we're finding complementary bases twice, which leads us back to the beginning. Does that make sense? I don't know, nobody's talking to me. Hopefully that makes sense. So the last step would be to find the amino acids that correspond to the tRNA codons. And on the left side, you can see I have an entire chart with like all the amino acids and what codes for these amino acids. So on the very left side, it says first letter. So you would look there first. For example, again, looking at the first codon, GAU and tRNA. So the first thing we do is look at G. So the row of G. On the top, you can see it says the second letter. Just tells us what column to look in. So we're in the G row, and since the second letter is A, we're in the A column. So it would be the third one on the bottom. Hopefully that makes sense. Third one on the bottom. And on the right side, on the very right side, it says third letter. And that just tells us in that little square which one of the 4 to look at. So the last letter is U. So our codon is GAU. That tells us to look at the last row, third column, first one in that square. Again, this is confusing. It would be so much nicer if I could like circle it, and but we can't do that. So we're making do with this. And that codes for, I think it's called aspirin. This only has the abbreviations of the amino acids because they're usually very long. Like there's tryptophan, alanine, valine, that kind of thing. It's long. So usually they'll just abbreviate it. If you're curious, you can go online and find like a complete list of like the full name of the amino acids. And each amino acid has like a specific function. Sometimes your doctor or someone might tell you to eat more foods that have like a specific amino acid. It's kind of like vitamins, but not really. You get what I mean, hopefully. And some amino acids can actually be produced by the body. So some can, some can't, which is why sometimes you need to eat more foods with like that specific amino acid. Anyway, something interesting to look at within the chart is if you look at the third row, first column, fourth one, it's in red, so that's actually easier to see. It says AUG MET. So this stands for methionine, or maybe it was methionine. I always pronounce methionine, but I don't know if I'm correct. And that is the start codon. So this just tells the RNA or mRNA or anything really where to start translating. And it's also like the first amino acid because that's where it starts. In this case, it's not because I just came up with some random DNA strand, but in real life it would be. And something else interesting, or the other interesting part, is if you look at the first row, in the last two columns, you can see that there are 3 codons, and next to it they say stop. So these are UAA, UAG, and UGA, and these are the stop codons. So this just means wherever that codon appears, the chain will stop. So at the very end of any protein chain or mRNA chain, that one of those will appear, and that will tell the amino acids to stop coming. The protein will leave the ribosome and then start folding. And that's why sometimes there will be mutations in DNA, because if there's some kind of mistake during the translation process and the protein stops early, then that can lead to some mutations in the organisms. We're not really going to talk about it right now because that's complicated. Another interesting thing, I guess, in the chart is you can see that sometimes multiple codons code for one amino acid, and that is again relating to relating to mutations. So since their DNA and this kind of stuff is so complicated, in order to prevent mutations from being too serious, a lot of times multiple things will code for one amino acid. So even if you mess up one base, even if you mess up one one base, it'll still have the same result because it codes for the same thing. And we are slightly running out of time. We only have 5 minutes left. So skipping slide 16 because that is just practice. If you want to, you can take a look at it and kind of the thought process through it. And just going straight to slide 17, which is DNA replication. So I'm going to try to go through this as fast and as simply as I can because it's complicated. So that's slide 17 is mostly just a diagram of it, so it's a bigger diagram, blown up diagram. But we're gonna mostly take a look at slide 18 because I have written down some of the more simple, broad steps. Okay, so first of all, if you look at the diagram, there is this blue triangle that is kind of pointing towards the right. That is called the helicase. The helicase unzips the DNA so that it can be replicated or duplicated or copied. And on a little more to the right, we see a green ring that is circular. That is called a Topo Isomerase. It took me a very long time to figure out how to pronounce it because honestly, how do you pronounce it? And that stops the DNA strand from supercoiling or just winding in general. So DNA is a double helix, so it's winding ladder, and in order to stop it from winding so that it can be properly replicated, topoisomerase is there. So the first step in DNA replication, obviously the helicase unzips it, and this forms 2 strands. There's a lagging strand, which is on the top in the diagram, and the leading strand in the bottom. And this is important because if you remember at the beginning, I said that the strands run anti-parallel to each other. Well, in DNA replication, the thing that comes in and actually provides the complementary bases, it can only move in one direction, which means that one strand, it's fairly simple, it's called DNA polymerase. So the DNA polymerase, if you look at the 4th step, the DNA polymerase comes in and fills in the complementary bases. So in the leading strand, it just goes straight. It can go just in one direction. However, in the lagging strand, it's slightly different because the strand goes in an opposite direction then the DNA polymerase moves. So I really wish I could draw. Okay, I'm gonna try my best. So if we had the I'm just going to say the lagging strand goes this way because it's simpler. Lagging strand goes to the right, then, and, okay, so I wish I could draw. Okay, I'm gonna try my best again. So let's just say, oh, we have questions. I'm sorry. Okay, sorry. First of all, Lokesh, I'm sorry if I'm pronouncing your name wrong. You are not unmuted. And this is a webinar, Zoom webinar, so that means you can only just like listen to me talk and ask questions. And I am not sharing my screen because unfortunately I am not host. Forensics will start in 2 minutes if I can finish this quickly. So I'm not sharing my screen because I'm not host. The monitor is, I think she's AFK, so I'm not host. I'm just talking through the presentation. I will share the link again at the end if, just right now. You know what, I'll send it. Again, I'll send the link, and that is the link to the presentation. So if you want to take a look, go ahead, because I'm talking through it. Anyway, continuing with the DNA replication. I'm sorry, I'm gonna go a bit over time, I guess. Okay, so the lagging strand.
Oh, it's fine. Just take as long as you want.
Thank you. Okay, the lagging strand, if it were to go to the right this way, then the DNA polymerase, it could go only go in this way. So okay, yeah, so the RNA, not the RNA, the DNA strand goes this way, but the DNA polymerase can only move this way. And let's just say RNA primers. Okay, so that just means, pretend this is a DNA polymerase. It just has to move like this, you know? And it moves in short bursts because it runs in the opposite direction, which means that it can't continuously create the bases. So that just leaves you with a bunch of small fragments, which are called Okazaki fragments. And later, the DNA ligase comes in at the very end, and it will seal up those fragments. After all this is done, you have 2 identical DNA strands. And earlier, there was a step I skipped because I was too busy talking about the lagging and leading DNA strands. But RNA primers are the purple thing. No, wait, that's not it. The RNA primers, it's kind of like a small, okay, think of it as like a starting flag, okay? So it comes onto the DNA strand on both of them, and it marks where to start. On the leading strand, it only marks it once because, again, the DNA polymerase can just move in a straight line. In the lagging strand, it marks like multiple start signs so that the DNA polymerase can like keep going around. And I think that is it. I do not want to go too over time. Any questions? I'm on the last slide, by the way. Any questions? I know this was very confusing. Obviously made it harder because I can't share my screen. If you have any remaining questions after this class, feel free to email me. And I do have homework. It's optional, I believe, if you want to do some extra practice or just like take a look at it. So, any questions before I leave?